So a pleasant day, dear doctors, and welcome to this module, this special coaching session, an intensive blitz in biochemistry. And we'll be taking up the pentose phosphate pathway, everything that has to be known about the pentose phosphate pathway, all of the high yield must-knows, and the things you have to bring to your upcoming examinations. So this is Doc 2, your med guru, and thank you for tuning in. So let's begin. So these are the following references which I will be using for this coaching session. I'll be using 3 very high yield biochemistry references, particularly Harper's Illustrated Biochemistry, Lippincott's Illustrated Biochemistry, and one of my favorites, Michael King's Integrative Medical Biochemistry. Now, if you plan to read from the mother textbook, then I suggest you read this reference on the chapter of the pentose phosphate pathway and other pathways of hexose metabolism. Now, let's talk about some basics about the hexose monophosphate shunt. Now, it has several aliases. It is also known as the pentose phosphate pathway, or what we commonly refer to as the PPP. It is also known as the phosphogluconic acid pathway, or the phosphogluconate shunt, and it is also known as Warburg-Dickens pathway. Now, for the sake of your examination, just remember HMP, or the hexose monophosphate pump, or the pathway, and PPP, which is the pentose phosphate pathway. Now, one of the significance of the hexose monophosphate shunt is it is an alternate pathway of glycolysis. So it is an alternate pathway for the metabolism of glucose. Now, there's something unique about the hexose monophosphate shunt compared to glycolysis, and that it does not generate ATP. So I repeat, it does not generate ATP. So 2 of the most important main functions of the hexose monophosphate shunt is to generate NADPH. Now, what is the significance of NADPH? This is utilized for fatty acid synthesis. And what is the second main function of the hexose monophosphate shunt? This is to supply or produce ribose 5-phosphate. This ribose phosphate is utilized as a substrate for nucleic acid synthesis. So it is involved in genetics, DNA, RNA. Now, of the several aliases that I mentioned earlier, these are the 2 aliases that recently popped out in the licensure exams. First is hexose monophosphate pathway or shunt. Next is phosphogluconate oxidative pathway. Okay, again, these are the 2 aliases that recently popped out. Hexose monophosphate pathway or shunt, and phosphogluconate oxidative pathway. Now, where is the hexose monophosphate shunt found? Where does this metabolic pathway occur? It occurs in the cytosol, and the rate-limiting enzyme is glucose 6-phosphate dehydrogenase. Now, this is commonly referred to as G6PD. Do not confuse this with the disease G6PD, because the disease is actually glucose 6-phosphate dehydrogenase deficiency. So again, these are two pearls that you have to bring with you to the upcoming examinations. Where is the site of metabolism for the hexose monophosphate shunt? It is found in the cytosol. What is the rate-limiting enzyme? It is the glucose 6-phosphate dehydrogenase. Now, question, can you give me the metabolic pathways that occur in the mitochondria? What are the metabolic pathways that occur in the mitochondria? Give me the metabolic pathways. So I believe I've taught students in the past this mnemonics, mito, fatto. So what is fatto? What is letter F? What is this letter F? This is fatty acid oxidation. What is letter T? This is TCA, or the Krebs cycle, and O is for oxidative phosphorylation. Everyone has to memorize this. Now, the next pearl I want everyone to remember is the connection, the link between these four important pathways. Glycolysis, which is the breakdown of glucose into pyruvate, if this is aerobic glycolysis, and the breakdown of glucose to lactate, if this is anaerobic glycolysis. Then we have the citric acid cycle, and we have the electron transport chain, which transfers electrons from complex 1 to complex 5 until ATP is produced. Here is the pentose phosphate pathway. It is an alternate pathway of glycolysis. I would like to use the analogy of navigation applications for driving, such as Waze or maybe Google Maps. So let's say, for example, you're passing through the road, which is called glycolysis. There is some traffic there. So what alternate route can you take? This navigation device will probably take you and tell you, instruct you, please pass, turn right, and pass through pentose phosphate pathway road. So this is an example of an analogy for alternative pathway. Now, how important is the pentose phosphate pathway? Now, there are actually three main hexoses, namely glucose. Glucose would be the most important. Fructose. Then we have galactose. So this pathway, okay, is for the metabolism of the main hexoses: glucose, fructose, galactose. There's interconversion of sugars that occurs, even if it's called pentose pathway. I will explain in a while this interconversion of sugars, which is the other function of the pentose phosphate pathway. Now, take note that these main hexoses are derived from dietary starch, sucrose, as well as lactose. So please take note of this. And these hexoses would be absorbed from the gastrointestinal tract. Now, the fructose and the galactose will be converted into glucose. And this conversion of fructose and galactose would basically occur in the liver. It would basically occur in the liver. Now, there are some disorders that arise from deficiencies of enzymes in the pentose phosphate pathway. Number one on the list is the genetic deficiency of glucose 6-phosphate dehydrogenase, or what we call G6PD. This is part of the newborn screening. Glucose 6-phosphate dehydrogenase is the first enzyme and the rate-limiting enzyme of the pentose phosphate pathway. Deficiencies in G6PD would result to hemolytic anemia. Other disorders would include essential pentosuria, galactosemia, hereditary fructose intolerance, and essential fructosuria. So essential pentosuria, essential fructosuria, galactosemia, and hereditary fructose intolerance. Now, always remember this, the reactions of the pentose phosphate pathway occur in the cytosol. Again, the reactions of the pentose phosphate pathway occur in the cytosol. So similar to glycolysis, the enzymes of this pathway are cytosolic. Now, if we discuss in detail the pentose phosphate pathway, you actually only have to know the following concepts for any standard biochemistry exam and for building or bringing with you the must-know basic foundations that can be applied in the clinical scenario. So first concept is that the pentose phosphate pathway is divided into two phases. Okay, the pentose phosphate pathway is divided into two phases. First is the oxidative phase. Second is the reversible non-oxidative phase. So the oxidative phase is irreversible. The non-oxidative phase is reversible. So the first phase is the irreversible oxidative phase. The second is the reversible non-oxidative phase. Now, let's talk about some details mentioned in the mother textbook. The first phase, which is the oxidative phase, involves a couple of reactions. This first involves the dehydrogenation of glucose 6-phosphate. After the dehydrogenation of glucose 6-phosphate is the decarboxylation. So memorize the two reactions in the first phase, dehydrogenation and decarboxylation. Now, always remember the product of the first phase is ribulose 5-phosphate. And once ribulose 5-phosphate is produced, the pentose phosphate pathway now proceeds to the second phase, which is the non-oxidative. The non-oxidative phase involves the conversion of ribulose 5-phosphate back to glucose 6-phosphate. So question, what product is similar to the first and the second phase? What product is similar to both the first and the second phase? In fact, it marks the end of the first phase and the beginning of the second phase. It is ribulose 5-phosphate. Okay, I want everyone to remember that. Okay, ribulose 5-phosphate. So here, ribulose 5-phosphate is the product of the first phase. It's also the beginning of the second phase. Now, the reactions of the pentose phosphate pathway involve mainly two enzymes. These two enzymes would be transketolase and transaldolase. So again, there are two enzymes involved. Okay, there are two enzymes involved in the pentose phosphate pathway, transketolase and transaldolase. So please take note of that. So here is a very, very busy illustration lifted from Mother Harper's. It begins with 3 molecules of glucose-6-phosphate. So here's the first molecule. Here's the second. Here's the third molecule of glucose-6-phosphate. So let's label this number 1, number 2, and number 3. There are 3 molecules of glucose-6-phosphate that give rise to 3 molecules of carbon dioxide and 3 5-carbon sugars. So what do we call 5-carbon sugars? What do we call 5-carbon sugars? We generally call 5-carbon sugars as pentoses. Okay, we call 5-carbon sugars as pentoses. Now, do you remember the first reaction that occurs? What is the first reaction that occurs in the pentose phosphate pathway? Is it dehydrogenation or is it decarboxylation? It is dehydrogenation. This is the dehydrogenation that occurs. And here is the enzyme glucose-6-phosphate dehydrogenase. Okay. Now, these 3 5-carbon sugars or pentoses will now rearrange. And basically, fast forward, there is going to be an intermediate that is going to be formed. This intermediate is going to be glyceraldehyde 3-phosphate. Okay, again, glyceraldehyde 3-phosphate. Here is the glyceraldehyde 3-phosphate that is going to be yielded or formed. Now, what are the 2 enzymes again, aside from glucose-6-phosphate dehydrogenase or G6PD, which are mainly involved in the pentose phosphate pathway? What are those 2 enzymes again? So number 1, we have transketolase. So here I'm labeling it right now. And we have transaldolase. Okay, transketolase and transaldolase. Now, question. Between these 2 enzymes, transketolase and transaldolase, which is thiamine containing? Which is thiamine containing? Which contains thiamine? Okay, it is transketolase. Okay, very, very important. Now, going back to this illustration, the second phase is the non-oxidative phase. So again, what is the end of the oxidative phase? What product is produced at the end of the oxidative phase? Okay, that product which is produced at the end of the oxidative phase is ribulose 5-phosphate. Look at this. I'm highlighting, I'm boxing out the ribulose 5-phosphate. Okay, and where does the second phase or the non-oxidative phase begin? It also begins with ribulose 5-phosphate. So in the second phase, which is the non-oxidative, ribulose 5-phosphate, okay, is going to be converted back to glucose-6-phosphate. 2 enzymes involved is going to be transketolase and transaldolase. Okay, please remember that. So take note that there are two phases. Now, this is very, very important. This is what we mentioned earlier, the first reaction. Here is glucose 6-phosphate. Here is the enzyme G6PD. So always remember the first step is dehydrogenation. This dehydrogenation of glucose 6-phosphate will yield what? What would the dehydrogenation yield? It would yield 6-phosphogluconolactone. Okay, so I want everyone to remember that. Any deficiency of this enzyme here, G6PD, would present clinically as hemolytic anemia. Hemolytic anemia. Now, in the non-oxidative phase, okay, again, in the non-oxidative phase, so we have ribose 5-phosphate, okay? Always remember ribose 5-phosphate would, ribose 5-phosphate converts, okay, this enzyme here, ribose 5-phosphate ketoisomerase, okay? Ribose 5-phosphate ketoisomerase will convert ribulose 5-phosphate into what? Ribose 5-phosphate, which is used for nucleic acid synthesis, nucleic acid and nucleotide synthesis. Is it ribulose or is it ribose 5-phosphate? Which is utilized, okay, for nucleic acid and nucleotide synthesis. Is it ribulose or is it ribose? It is ribose 5-phosphate. Okay, please remember that. Now, bring this with you to the board exams. Ribulose 5-phosphate is converted to ribose 5-phosphate by what enzyme? It is ketoisomerase, the ribose 5-phosphate ketoisomerase. Why is this important? Because it is ribose 5-phosphate that is used for nucleic acid synthesis. So bring this with you to the board exams. Okay, now I also want you to bring this to the board exam. Glycolysis and pentose phosphate pathway have some similarities, but they are actually different from each other. So in this table here, okay, again, in this table here, I want you to take a look that both occur in the cytosol. Okay, glycolysis and pentose phosphate pathway both occur in the cytosol. This is the first difference you have to memorize. Rate-limiting enzyme for pentose phosphate pathway is G6PD. Again, rate-limiting enzyme for the pentose phosphate pathway is G6PD. Rate-limiting enzyme for glycolysis is phosphofructokinase or PFK. Please remember, pentose phosphate pathway does not produce ATP. It does not produce ATP. So memorize that by heart. Now, one of the major functions of the hexose monophosphate shunt, one of the main functions of the hexose monophosphate shunt is to produce NADPH. So I want everyone to remember the moment you hear pentose phosphate pathway, think of NADPH. This NADPH is very important because it is utilized for the biosynthesis of both nucleic acids, steroids, and fatty acids. but the role of NADPH is significantly more important in fatty acids and steroids. NADPH is also used for the supply of reduced glutathione, and this reduced glutathione serves to protect particularly the RBCs from reactive oxygen species and oxidative stress. NADPH also plays a very important role in the bactericidal activity of polymorphonuclears or neutrophils. NADPH also takes part in the cytochrome P450 system in the liver. Now, most important clinical correlate for the functions of NADPH. It's glutathione, and it is the bactericidal activity. What disease arises when there is reduced glutathione? So if there is a lack of G6PD, such as in G6PD deficiency, no glutathione results to hemolytic anemia. And if there is a problem with the bactericidal activity of the polymorphonuclears, this will lead to chronic granulomatous disease. They will be prone to recurrent pyogenic infections. Now, this is what will come out in your exam. The role of the pentose phosphate pathway and glutathione peroxidase in protecting the RBC against hemolysis. So always remember, in the red blood cell, the pentose phosphate pathway is the sole source of NADPH. NADPH is required for glutathione. It is the reduced glutathione or the reduced form, okay? This is glutathione reductase, which removes hydrogen peroxide. There is a very important mineral that glutathione peroxidase, the enzyme, contains, and that is selenium. What fat-soluble vitamin is important for glutathione metabolism? What fat-soluble vitamin is important or essential for glutathione metabolism? It is vitamin E. Okay, vitamin E. Now, in this figure from the mother textbook, it shows you the pentose phosphate pathway is the sole source of NADPH in the RBC. This is required by the enzyme glutathione reductase, because the reduced form is the one. The reduced glutathione is what protects the RBC from oxidative stress. Now, question. This recently popped up in the boards. What observation states that cancer cells tend to ferment glucose into lactate even in the presence of sufficient oxygen to support mitochondrial oxidative phosphorylation? What pathway is oxidative phosphorylation? Where will you encounter it? You will encounter this in the electron transport chain. So what is this observation? Okay, that states that cancer cells tend to ferment glucose into lactate even in the presence of sufficient oxygen. Remember, lactate production is supposed to be under anaerobic scenarios. So this is the famous Warburg effect. Yes, Warburg metabolism is actually correct. Now, this was discovered by Warburg. This is the reason why. What is the other alias of the pentose phosphate pathway? What is the other alias of the pentose phosphate pathway? It is also known as the Warburg-Dickens pathway. This is how the mother textbook helps you top the exam. Okay, so please take note of this. Now, this one also came out very recently. What is this pathway wherein there is the formation of 2,3-BPG or 2,3-bisphosphoglycerate, which regulates the release of oxygen from hemoglobin such that it will be delivered to the tissues. What do we call this pathway? Okay, this is the famous Rappaport-Lubering pathway, also known as the Rappaport-Lubering shunt. Okay, please take note of this. And please take note of this. Rappaport-Lubering pathway. Now, this is the Rappaport-Lubering pathway, which is actually found in the mother textbook, but they do not use the term Rappaport-Lubering. They use the term 2,3-bisphosphoglycerate pathway. So 2,3-BPG pathway. So let's end up with the clinical correlates for this coaching session. First and foremost, and very predictable in the exam, is glucose 6-phosphate dehydrogenase deficiency. Please remember, this is an X-linked recessive disorder. Now, being an X-linked disorder, what is the sex or the gender of the patient? If this is X-linked, who will be symptomatic? Who will be symptomatic? Sino yung magkaroon ng sakit? Who will be sick? It is the male, because this is X-linked. Okay, this is X-linked male. So what are the manifestations of G6PD deficiency? Number one on the list is hemolytic anemia. They can also present with manifestations of chronic granulomatous disease. So they are going to present with hemolysis. Now, always remember, female heterozygotes of G6PD have increased resistance to malaria. Now, pharmacology crossover. What are the drugs which are contraindicated in patients with G6PD? What very important drugs are contraindicated? Okay, if we go by classifications, it would be anti-malarials. Okay, that would include primaquine, quinidine, okay, anti-malarials, as well as sulfonamides. So please memorize these two drugs, anti-malarials and sulfonamides. So please take note of this. Now here is basically a table of the drugs which have definite risk of hemolysis in patients with G6PD deficiency. Anti-malarials, I want you to memorize first. sulfonamides next. You can probably add the antihelminthics next. Okay. Always remember you're preparing for a multiple choice exam with 4 to 5 choices. You don't have to memorize everything. Now, the second correlate is favism. Now, what you see here in the photo below are the fava beans. These are the broad beans, which are part of the Mediterranean diet. So in patients with glucose 6-phosphate deficiency, when they ingest favism, there is a hemolytic crisis that occurs within 24 to 48 hours after ingestion. They present with pallor, hemoglobinuria, jaundice, and severe anemia. So please take note of this. So pallor, hemoglobinuria, jaundice, and severe anemia. So this is favism. Then other disorders mentioned in the textbook. So we mentioned the G6PD already. So intake of antimalarials, fava beans, hemolytic anemia. Then we have the Wernicke-Korsakoff, which is associated particularly in what population of patients? What populations would usually be developing Wernicke-Korsakoff encephalopathy? It is usually the alcoholics. So this is thiamine deficiency or vitamin B1 deficiency. What is that enzyme again, which contains thiamine? That very important enzyme, which plays a role in the pentose phosphate pathway, it is trans-ketolase. Okay, trans-ketolase. So in Wernicke-Korsakoff, there is deficient trans-ketolase activity. Now, I would like to share this with you, this famous mnemonics code rack. So Wernicke's encephalopathy is different from Korsakoff psychosis. So in Wernicke's encephalopathy, it presents with confusion, ophthalmoplegia, and ataxia. That is the triad. Okay, that is the triad. Okay. That is the triad of Wernicke's encephalopathy. So confusion, ophthalmoplegia, ataxia, and thiamine deficiency. Confusion, ophthalmoplegia, ataxia. This is seen in thiamine deficiency. Now, once there is amnesia, whether retrograde or anterograde, plus confabulations, this is now Korsakoff's psychosis. Now, question. Let's see who's going to top these boards. What specific part of the brain is affected in Wernicke-Korsakoff psychosis? What specific part of the brain is affected most in Wernicke-Korsakoff psychosis? It is the mammillary bodies. Okay, the mammillary bodies. Okay, so please remember that, the mammillary bodies. Great. Now, winding up with the clinical correlates, we have chronic granulomatous disease. So as mentioned, this is caused by a genetic deficiency of NADPH oxidase. This is the enzyme you need to memorize. NADPH oxidase. Now, these individuals have defective neutrophils. They are prone to recurrent pyogenic infections and infections caused by catalase-positive organisms. We usually perform the nitroblue tetrazolium tests, and it would be negative in patients with CGD. They also present with granulomas of the skin, the scarring, like what you see here, hence the term granulomatous disease. So enzyme, NADPH oxidase. Now, for us to top the exam, we need to dig all of the references which are found in the syllabus of the Board of Medicine. They mention disorders of fructose metabolism. So in hereditary fructose intolerance, there is a deficiency of aldolase B. This is an autosomal recessive disorder which presents with hypoglycemia and vomiting after ingestion of fructose-rich foods. So hypoglycemia and vomiting after ingestion of fructose-rich foods. Now question, what is the name of the fruit tree which contains hypoglycine, the toxin hypoglycine? What is the name of the fruit tree which is mentioned in Harper's Biochemistry? It is the ackee tree. So the unripe fruit of the ackee tree has the toxin called hypoglycine. So there is significant hypoglycemia. What is the problem in this disease, which we call Jamaican? Can you fill in the blanks? Jamaican vomiting sickness. What is the pathophysiology of Jamaican vomiting sickness? Is it a problem with glucose metabolism? Is it a problem with fructose metabolism? Is it a problem with the pentose phosphate pathway? Is it a problem with glycolysis? What is the problem in Jamaican vomiting sickness? It is a problem with the beta oxidation, okay, of the fatty acids. A beta oxidation of the fatty acids, particularly the enzyme acyl-CoA dehydrogenase. Acyl-CoA dehydrogenase. Now, winding up, another disorder we must remember is Essentialfructosuria. This is because of the deficiency of the enzyme fructokinase, a hepatic fructokinase. It presents with fructosemia, so fructose found in the blood, and fructosuria, fructose found in the urine. So this is essential fructosuria. So I hope everyone takes note of this. Now, question, what glycogen storage disease has a deficiency of the enzyme phosphofructokinase? What glycogen storage disease has a deficiency in the enzyme phosphofructokinase. Okay, what GSD type is this? Glycogen storage disease type 7, also known as Tarui's disease. Okay, that's Tarui's disease. So please remember that. Von Jerk's disease is type 1. Okay, that's glucose 6-phosphatase. So do not mix them up. Now, as we end this module, I need to remind you for your examinations regarding the disorders of galactose metabolism. So number 1 on the list, and this is the only thing you have to bring with you, it's galactosemia. It can either be a deficiency of galactokinase or a deficiency of galactose uridyl transferase. Some would refer this to as GALT. So these are the 2 enzymes deficient that can be deficient in galactosemia. So these babies would present with cataracts. They also present with hepatosplenomegaly. There's going to be hepatic failure. Okay, the cataracts for me is really a standout for you to memorize. Okay, the cataracts. So lastly, this is a photo from the galactosemia support group. They can present with cerebral edema. They can present with jaundice. They can present with vomiting, failure to thrive. They present with renal failure. They present with liver failure. Okay, it's really the liver failure which causes demise once they develop jaundice, and they are prone to sepsis. So these are the common manifestations of galactosemia. The one I predict for your exams is the cataract. The presence of the cataracts. So I hope you learned something. Okay, I hope you learned something from this coaching. Okay, this utilized 3 different very high yield, the most high yield references for any licensure exam, for any biochemistry exam. It is very textbook based, it's syllabus based. So we can't get any more high yield than that. So this is Dr. saying thank you very much for listening.